I'd now like to welcome Tom Ashton to the stage, who is the Technical Sales Manager from Fermenters. He's here to talk about a new yeast strain from Fermenters. It's not quite yet available, but is very exciting because it makes hoppy lagers, so don't anyone go anywhere. It is called, I knew I'll get the numbers wrong, SH-45. All right, thanks, Tom.
Thanks, Will. Before I kick off, thanks again to Bintani for hosting today. It's been amazing to catch up with people and celebrate their 30th anniversary, 30th birthday. And thank you all for coming out today as well. Thanks for the introduction, Will. So my name's Tom. I'm the Technical Sales Support Manager for Fermenters. Also here with me today, I've got Amanda, who's our Sales Manager for Australia and New Zealand. Amanda, if you want to stand up and say hello. If you want to... after my presentation today, if you want to come grab us and have a chat about anything, you're more than welcome to. So today I'm talking about our new SafLager SH-45 strain, which is our latest yeast solution to elevate aromas in lager beers. Hop aromas, I should say. So today I'll give a brief introduction into lager yeast. We'll then look at SafLager SH-45 compared to our other lager yeast strains. And then I'll go into a bit more technical stuff involving biotransformation and biotransformation of SafLager. And then we'll wrap it up. So taking it back to the basics, looking at kind of the two main classic styles of yeast. So we broadly classify them into two different ranges. We've got the lagers, which are traditionally referred to as kind of bottom fermenting beers, which are generally fermented at cooler temperatures. Your ales are considered top fermenting, fermenting at warmer temperatures. So the technical name for lager strains, Saccharomyces pastorianus, Saccharomyces cerevisiae for ale. So what is Saccharomyces pastorianus? Well, it's a hybrid. And a hybrid is when two different species breed together to give an offspring. So the hybrid for pastorianus starts with cerevisiae, which is pretty much ubiquitous. So cerevisiae has been around for as long as beer and wine has been around, so they've been around for millennia. Given that time, Saccharomyces cerevisiae has had plenty of time to kind of evolve, mutate, and that's why we have such a broad range of different ale strains available now. You know, from your German Hefeweizens to your different types of Belgian beers, to your American beers, to your British beers, etc. So where the hybrid that came from for it was cerevisiae and eubayanus, and its possible origin was in Patagonia. So these two yeasts are thought to have naturally hybridized. It's thought to have probably occurred in a natural environment such as the brewery. And that's led to the development of pastorianus. So this probably happened around the 17th century. So compared to how old like cerevisiae is, it's still relatively recent. And because it's relatively recent, that's why you see, in general, a kind of a less diversity in lager strains compared to ale strains. So what are the main properties of Saccharomyces pastorianus? So they have ability to ferment at lower temperatures, generally maltotriose positive. They're POF negative, and they generally have pretty good sedimentation and flocculation. So looking at Fermenters' SafLager yeast range currently, we've got 4 different offerings, ranging from most popular, which is the W3470, which is the famous Weihenstephan lager strain, reliable, neutral. And that kind of ranges from all the way up to our most recent release, which was the E30, which has produced a slightly more estery, slightly more isoamyl acetate, more kind of European continental kind of lager. So now introducing SH-45. And so to do this, once we've developed we've selected the yeast strain that we're going to develop, we then put it through a set of experimental conditions so we can kind of categorize it. And we generally do this in kind of a standard set of conditions depending on the yeast involved. So here we have standard wort, so we usually use a 15 Plato wort with just straight Pilsner malt, standard pitching rate of 100 grams per hectoliter. fermentation of 14 degrees Celsius with a 14-day maturation time followed by filtration. So these experiments were all conducted in a laboratory scale. And so what we found, again, apologies if you're at the back, you probably can't see this particularly well, I will explain it. What we did find was that the main differences were the kind of lag phase of different strains. And so E30, which is the kind of brownish color one that has, you can kind of see as the flattest kind of starting after 2 days, has the longest lag phase. However, the main fermentation period, you can see they've all got pretty similar kind of fermentation rates after that kind of 48-hour mark. And actually, after kind of seven or 8 days, at the end of once they've reached attenuation, they all reach attenuation at about the same time and at about the same attenuation amount. Looking at attenuation, all pretty similar. So SH-45, approximately 82%, so pretty much in line with our other strains. Ester production. When we're talking about esters have different flavor thresholds, so it's quite hard to say, or it can be misleading to say. If we look at ethyl acetate, which is the predominant ester produced by Saccharomyces cerevisiae and also Saccharomyces pastorianus, you'll see that ethyl acetate is way up there in terms of parts per million, but actually ethyl acetate has a much higher aroma threshold. So what we do is we divide the measured concentration of that compound by the aroma threshold to give a kind of an odor unit. So basically, if the odor unit is more than 1, it means it's probably or possibly perceivable in that beer, and therefore anything less than 1 is unlikely to be perceived. So you can see with our standard S23, S189, and W3470, under these standard conditions, they all produce esters below, just below or just on the flavour threshold. Compare that to E30, which I mentioned before, has got slightly higher kind of isoamyl acetate, slightly higher kind of banana fruity characteristic. You can see that's above the threshold, but not by much. In these conditions, an aroma threshold as 2 is perceivable, but not outrageous. And SH-45, slightly higher esters compared to the S23, S189, and W3470. However, only just. And just the only one there is isoamyl acetate at approximately 1.4. When we put it through our sensory panel, so this is our sensory panel at our Fermentis campus, highly trained sensory panel, all done blind. And they were given this beer and with different flavour attributes to kind of score it against. We saw it scored very high. This is a slightly different beer base. So compared to the other previous one, this one's a 14 Plato wort. And it's fermented at 14 degrees and had a free rise up to 17 degrees at 50% attenuation. And then it was dry hopped on day 2 with a combination of Citra, Amarillo, and Hallertau Blanc. And what we found is a really good expression of citrus and tropical, and also thiolic kind of character. So thiolic in this case has been that kind of Sauvignon kind of gooseberry character. So obviously this is quite a new strain. There's still more trials and experiments to come. However, from a sensory point of view, showing really good potential. So finally, we'll look at the biotransformation potential of the SafLager SH-45, which is probably the reason that it got chosen for this application. So when we talk about hops, we can talk about the kind of the hidden potential of hops. So obviously you've got the hop oils, but you've also got thiols, you've got terpenes as well. So there's two main families. Thiols can be described as passion fruity, citrusy, peachy. However, they are unperceivable when they're bound to an amino acid such as cysteine or glutathione. And the ratio between the free thiols and the bound thiols depends on the hop variety. Compare that to terpenes, which is kind of more floral, rose-like, or citrusy. These can be bound to sugar chains, which makes them unperceivable. And again, the ratio also depends on the hop variety itself. So when we're looking at thiols, so we have I think the thiol in this picture is looking at, they've got the fruit that's kind of bound to the amino acid. And when it's bound like this, the flavor threshold is significantly higher. So in this case, we're talking about the amino acid cysteine, which we can call cis-thiol for short, or a cis-adduct. Or it can be glutathione, which we can also refer to as G-thiol or GSH adduct. So in the presence of certain yeast, and yeast expressing certain enzymes, these enzymes can then cleave that bond, releasing that thiol, and then the flavor of that specific thiol can be released and then perceived. The reason for this is that the free unbound thiol has a much lower flavor threshold compared to the bound thiol. And we're talking about thresholds, in some cases, we're talking in nanograms per liter, so very, very low. There's two main formats or two main structures of the thiols that we generally refer to. We refer to them either as the alcohol form. So we've got kind of three sets of examples here. The sulfanyl-hexanol, or sulfanyl-hexyl acetate. So you've got the alcohol form and you have the acetate form. You can see that based on the structure. And depending on whether it's the alcohol form or the acetate form, it can have slightly different aromas as well, and also different thresholds. So looking at the first one, for example, you can look at 3-sulfanyl-hexyl acetate has a threshold of 5 nanograms per liter. Compare that to the alcohol form, which is 55. And so we're referring to nanograms per liter. Like, what does that mean? So a nanogram per liter is not parts per billion; it's parts per trillion. So we can compare that to, say, like 4VG, which is the classic phenol in your German Hefeweizenbeers. So that has an aroma threshold of approximately 300 micrograms per liter, which is 300 ppb. Which we can kind of picture as being 6 drops in a jacuzzi of 1,000 liters. You compare that to 5 nanograms per liter, which is 5 ppt, parts per trillion. We're talking about 6 drops in an Olympic-sized swimming pool, and 4 million liters. So it's these are very, very flavor active, and it's quite impressive when you kind of compare it on that scale. So what is the metabolic pathway? So you've got the standard metabolic pathway here of glucose fermentation, producing ethanol, as well as some esters such as ethyl acetate, some higher alcohols as well. But when you have the thiol precursor, and here we're using the example of 3-sulfonyl-hexanol, comes into the yeast cell, and the asterisk there is referring to enzyme activity. So enzyme activity for a cysteine-bound thiol is beta lyase, or if it's a glutathione-bound thiol, it's a combination of different enzymes. So glutathione is a 3 amino acid chain, so it requires a few more enzymes, an extra step to kind of break that out and cleave the thiol from the amino acid group. That is then expressed outside the cell as its either alcohol or acetate form. So here's the study that we did looking at SH-45 and its ability to release these free thiols. And so in this experiment, we added in a cis adduct. So this is a cysteine-bound thiols at 10 ppm, and also glutathione adducts a little bit more because it requires a few more. The release is not as much because you have to, again, have extra enzyme activity to cleave it. So that was added at 15 ppm. It was fermented with unhopped wort, so no other influences from any other hops. Fermented it 7 days at 24 degrees, matured at 3 days at 4 degrees, and then the thiol was then extracted and then analysed. So what do we find? So on the top graph on the left-hand side, we've got our SafAle strains. These are our POF negative SafAle strains, and on the right-hand side, SafLager. So the top graph is the cysteine thiol release, and the bottom is the glutathione thiol release. So what we see is our K97, which is our traditional Col strain, which we've done a few studies on, that has shown the best application for thiol release for cysteine bonded thiols. You can see that's across the board, across both ales and lagers. K97 still remains as our best potential for releasing cysteine-bound thiols. However, interestingly, if we look at the glutathione thiols, so this is the one that requires more than just beta lyase, this is other enzymes as well to cleave. We see that all the SafAles are quite poor. So they are quite poor at this release, potentially they don't express those same enzymes. Compare that to the lagers. Across the board, lagers generally are better at cleaving those glutathione thiols, and especially SH-45. And looking at the dark color, so the kind of brownish bar represents the alcohol form, and then the kind of more cream beige color is the acetate form. And remember from previous slides, the acetate form is the more highly active, flavor active. So that's also indicating some very good kind of aromatic potential for this yeast. So looking at the potential of a couple examples of some various hops. So this is looking at without esterification. So on the left-hand side, you've got the three thiols in the hops. You've got the green bar, look at Hallertau and Citra, is the acetate form of the 3-sulfanyl hexanol, and then the 3-sulfanyl hexyl acetate, I should say, and then you can see just in the Citra at about 0.9 odor units again. So sorry, going back, odor units again is the amount that's measured over the aroma threshold. So you can see Citra's got a small amount, however, it's below the aroma threshold at about 0.9 odor units for the alcohol form. Going over to the odor unit potential for the cis and cysteine and glutathione precursors. So these are the thiols that are bonded to these amino acids. You can see the odor units there are in the hundreds or in the thousands. So there's plenty of these thiols that are present in the hops but are bound to the cysteine or glutathione. So they're there, but they're not being released, so they're not expressing any of these very highly aromatic flavor potentials. So when we're looking at to kind of understand and interpret the results of the next kind of slide, we're talking about thiol release ratio. That is the percentage of release equals the parts per million of freed thiol divided by the parts per million of the adduct. And then odor units, which I've introduced before, is the parts per million of the 3-thiol divided by the thiol threshold. And as mentioned before, as with the swimming pool analogy, thiol threshold is very, very low. So even a very small amount it can have a major impact. So what do we find? So the expression of this is again in odor units of Citra for a start. So this is from cysteine and glutathione adducts at a dry hopping rate of 400 grams per hectoliter. On the left-hand side, you had the alcohol form, so 3SH alcohol. In the yellow cream kind of color is the free thiol that exists within the hop. And then the reddish brown represents the released thiol from the yeast. So you can see that as a baseline in Citra, there's only a small amount of either free. There's only a small amount of free thiol from the actual hop itself. But in the acetate form, it's already contributing about 20 odor units. But as you can see, the SH-45 then releases a further 25 to bring it up to approximately 45. This is another example of Hallertau Blanc, with again quite a small amount of the alcohol fraction, with some, still in SH-45, there's still a significant amount released, and is more than an odor unit of 1. We're still looking at an odor unit of approximately 3 or 4 on that graph. However, on the acetate version, again, the lower threshold, you see a much more active, so releasing up to 25 odor units, which is much more significant compared to the other lager strains. And then finally, looking at Amarillo. Amarillo, if we go back a few slides, Amarillo in the left-hand graph, you got basically 0 free thiols in the actual hop itself, and you can see in the odor potential on the right-hand side is moderate to significant. And then we kind of jump forward. You can see that the baseline of free thiols is 0 for both the alcohol and the acetate form. So all the thiols measured here is coming from the yeast and enzyme activity. And again, SH-45 compared to the other lagers is the best performer in terms of biotransformation. So in general, key learnings from SH-45. It's a medium, I'd say probably medium-high attenuating in the low 80s. We found that it ferments really well at 14 degrees Celsius. Fully assimilates maltose, partly maltotriose. Works well at a pitching rate of approximately 100 grams per hectoliter, which gives about an ADF of about 82% on standard wort. Produces a low level of kind of that fruity estery character, again, depending on the fermentation condition. But the key aspect here is that significant release and esterification into the acetate form of those the glutathione thiols. So we can build on our SafLager yeast range now. SH-45 on the bottom, and so it's medium fruity estery, and with the key one being the thiol biotransformation property. So it's the perfect yeast to enhance that thiol release for hoppy lager beers. So new SH-45 shows fermentation speed that's similar to the other lager strains, and it has a really good ability to release those free thiols from glutathionate precursors, which is kind of compared to the SafAle strains, which are really good for the cysteine ones. And the biotransformation potential from the hops from the glutathione precursor is much higher than compared to cysteine. So yeah, lastly, a knowledge of Louvain Institute, which helped us perform these studies. And lastly, thank you again. Thanks for coming out, and yeah, happy birthday again to Bintani. Yeah, any questions?
Any questions?
If not, yeah, come grab myself or Amanda for a chat later on. So as I mentioned before, SH-45 production is underway. The exact release date, not sure yet. Obviously, production is done at our facility in Belgium, and then once it goes through its quality control, will then have to be shipped out to us here in Australia, which takes a little bit more time. So second half of the year, look out for it. Bintani will send some comms later on. And yeah, if you got any questions, reach out to Amanda or the Bintani team. We'll help you out with that. So yeah, thank you very much.